Class 11, Chapter 9 Strategies for Enhancement in Food Production. With ever increased population of the world, enhancement of the food production is a major necessity. Biological principles as applied to animal husbandry and plant breeding have a major role in our effort to increase food production. Several new techniques like embryo transfer technology and tissue culture techniques are going to play a vital role in further enhancing food production. 9.1 Animal husbandry. Animal husbandry is the agricultural practice of breeding and raising livestock. As such, it is vital skill for farmers and is as much science as it is art. Animal husbandry deals with the care and breeding of livestock like buffaloes, cows, pigs, horse, cattle, sheep, camel, goat, etc. that are useful to humans. Extended, it includes poultry farm and fisheries. Fisheries include rearing, catching, selling, etc. of fish, mollusks, shellfish, and crustaceans, prawn, crabs, etc. Since time immemorial, animals like bees, silkworms, prawns, crabs, fishes, birds, pigs, cattle, sheep, and camels have been used by humans for products like milk, egg, meat, wool, silk, honey, etc. It is estimated that more than 70% of the world livestock population is in India and China. However, it is surprising to note that the contribution to the world farm produce is by 25%. The productivity per unit is very low. Hence, in addition to conventional practice for animal breeding and care, newer technologies also have to be applied to achieve improvement in quality and productivity. 9.1.1 Management of farms and farms animals. A professional approach to what have been traditional practice of farm management gives the much needed boost to our food production. Let us discuss some of the management procedures employed in various animals farm system. 9.1.1.1 Dairy farm production. Dairying is the management of animals for milk and its product for human consumption. Can you list the animals that you would expect to find in a dairy? What are the different kinds of products that can be made with milk from a dairy farm? In dairy farm management, we deal with processes and systems that increase yield and improve quality of milk. Milk yield is primarily dependent on the quality of breed in farm. Selection of good breeders having high yielding potential under the climatic condition of the area combined with resistance to disease is very important. For the yield potential to be realized, the cattle have to be well looked after, they have to be housed well, should have adequate water, and be maintained disease free. The feeding of cattle should be carried out in a scientific method with special emphasis on the quality and quantity of fodders. Besides stringent cleanliness and hygiene, both of the cattle and the handlers are of paramount importance while milking, storage, and transport of the milk and its product. Nowadays, of course, much of these processes have become mechanized, which reduce chance of direct contact of the produce with the handler. Ensuring these stringent measures would, of course, require regular inspections with proper record keeping. It would be help to identify and rectify the problem as earlier as possible. Regular visits by a veterinary doctor would be mandatory. You would probably find it interesting if you were to prepare a questionnaire on diverse aspects of dairy keeping and then follow it up with a visit to a dairy farm in your locality and seek animals to the questions. 9.1.1 2. Poultry farm management. Poultry is the class of domesticated fowl birds used for food or for their eggs. They typically include chicken and ducks and sometimes turkey and geese. The word poultry is often used to refer to the meat of only these birds, but in a more general sense it may refer to the meat of other birds too. As in dairy farming, selection of the disease-free and suitable breed, proper and safe farm condition, proper feed and water and hygiene and health care are important component of poultry farm management. You may have seen TV news or read newspaper reports about the bird flu virus, which created a scare in the country and drastically affected egg and chicken consumption. Find out more about it and discuss whether the panic reaction was justified. How can we prevent the spread of the flu in case some chicken are infected? 9.1.2 Animal breeding. Breeding of animals is an important aspect of animal husbandry. Animal husbandry aims to increase the yield of animals and improving the desirable quantities of the produce. For what kind of character would we breed animal? What would the selection of character differ with the voice of animals? What do we understand by the term breed? A group of animals related by descent and similar in most character like general appearance, features, size, configuration, etc. are said to belong to a breed. Find out the names of some common breed of cattle and poultry in the farm of your area. When breeding is between animals of the same breed, it is called inbreeding, while crosses between different breeds are called outbreeding. Inbreeding refers to the mating of more closely related individuals within the same breed for six generations. The breeding strategies is as follows. Superior males and superior females of the same breed are identified and mated in pair. The progeny obtained from such mating are evaluated and superior males and females among them are identified for further mating. A superior female in the case of cattle is the cow or buffalo that produce more milk per lactation. On the other hand, a superior male is a bull which gives rise to superior progeny as compared to those of the other males. Try to recollect the homozygous pure line developed by Mendel as discussed in chapter 5. A similar strategy is used for developing pure line in cattle as was used in case of bees. Inbreeding increases homozygosity. Thus, inbreeding is necessary if we want to evolve a pure line in any animal. Inbreeding exposes harmful recessive genes that are eliminated by selection. It also helps in accumulation of superior genes and elimination of less desirable genes. Therefore, this approach, where there is selection at each step, increases the productivity of inbreed population. However, continued inbreeding, especially close inbreeding, usually reduce fertility and even productivity. This is called inbreeding depression. Whenever this becomes a problem, selected animals of the breeding population would be mated with unrelated superior animal of the same breed. This usually helps restore fertility and yield. Outbreed is the breeding of unrelated animals which may be between individuals of the same breed but having no common ancestor for 4 to 6 generations. Outcrossing or between different breed, crossbreeding or different species, interspecific hybridization. Outcrossing is the practice of mating of animals within the same breed but having no common ancestor on either side of their pedigree up to 4 to 6 generations. The offspring of such a mating is known as the outcross. It is best breeding method for animals that are below average in productivity in milk production, growth rate in beef, cattle, etc. A single outcross often helps to overcome inbreeding depression. Crossbreeding. In this method, superior males of one breed are mated with superior females of another breed. Crossbreeding allows the desirable quantity of two different breeds to be combined. The progeny hybrid animals may themselves be used for commercial productivity. Alternatively, this may be subject to some form of inbreeding and selected to develop new stable breed that may be superior to the existing breed. Many new animal breed have been developed, but by this approach, Hazardale is a new breed of sheep developed in Punjab by crossing Bicanary Eves and Merino Ram. Interspecific hybridization. In this method, male and female animals of two different related species are mated. In some cases, the progeny may combine desirable features of both the parents and may be of considerable economic value. Example, the mule, figure 9.2. Do you know what cross lead to the production of the mule? Controlled breeding experiments are carried out using artificial insemination. The semen is collected from the male and is chosen as a parent and injected into the reproductive tract of the selected female by the breeders. The semen may be used immediately or can be frozen and used at the later date. It can be transported in a frozen form to where the female is housed. In this way, desirable mating are carried. Artificial insemination help us overcome several problems of normal mating. Can you discuss and listed some of them? Often the success rate of crossing mature male and female animals is fairly low even though artificial insemination is carried out. To improve chances of successful production of hybrids, other means are also used. Multiple ovulation embryo transfer technology (MOIT) is one such program for herd improvement. In this method, a cow is administered hormones with FSH-like activities to induce follicular maturation and superovulation. Instead of one egg, which they normally yield per cycle, they produce 6 to 8 eggs. The animal is either mated with an light bull or artificial inseminator. The fertilized egg at 8 to 32 cell stage are recovered non-surgically and transferred to surrogate mother. The genetic mother is available for another round of superior ovulation. This technology has been demonstrated for cattle, sheep, rabbits, buffaloes, maize, etc. High milk yielding breeds of females and high quality lean meat with less lipid meat. Yellow bulls has been bred successfully to increase herd size in a short time. 9.1.3 Beekeeping. Beekeeping or apiculture is the maintenance of hives of honeybee for the production of honey. It has been an age-old cottage industry. Honey is the food of high nutritive value and also find use in the indigenous system of medicine. Honeybee also produce beeswax, which find many use in industries such as in the preparation of cosmetic and polishes of various kinds. The increased demand of honey has led to large-scale beekeeping practice. It has been an established income-generating industry, whether practiced on a small or on a large scale. Beekeeping can be practiced in any area where there is sufficient bee pasture of some wild shrubs, fruits, orchard, and cultivated crop. There are several species of honeybees which can be reared. Of these, the most common species is Apis indica. Beehives can be kept in one's courtyard, on the veranda of the house, or even on the roof. Beekeeping is not labor intensive. Beekeeping, though relatively easy, does require some specialized knowledge, and there are several organizations that teach beekeeping. The following points are important for successful beekeeping. First, knowledge of the nature and habits of bees. Second, selection of suitable location for keeping the beehives. Third, catching and hives of swans, group of bees. Fourth, management of beehives during different seasons. And fifth, handling and collection of honey and of honey wax. Bees are the pollinator of many of our crop species. See chapter 2, such as sunflower, brassicacia, apple and pear. Keeping beehives in crop fields during flowering period increase pollination efficiency and improve the yield, beneficial both from the point of view of crop yield and honey yield. 9.1.4 Fisheries. Fisheries is an industry devoted to the catching, processing or selling of fish, shellfish or other aquatic animals. A large number of our population is dependent on fish products and other aquatic animals such as prawns, crabs, lobsters, edible oyster, etc. for food. Some of the freshwater fishes which are very common include cattleya, rohu, common crab. Some of the marine fishes that are eaten include hilsas, sardines, mackerels, and pomfrets. Find out what fishes are commonly eaten in your area. Fishes has an important place in Indian economy. It provide income and employment of millions of fishermen and farmers, particularly in the coastal states. For many, it is only source of their livelihood. In order to meet the increased demand on fisheries, different techniques have been employed to increase production. For example, through agriculture and pisciculture, we have been able to increase the production of aquatic plant and animals, both freshwater and marine. Find out the difference between pisciculture and agriculture. This has led to the development of and flourishing of the fishery industry, and it has brought a lot of income to the farmers in particular and the country in general. We now talk about blue revolution as being implemented along the same line as green revolution. 9.2 Plant breeding. Traditional farming can only yield a limited biomass as food for humans and animals. Better management practice and increase in acreage can increase yield, but only to a limited extent. But plant breeding as a technology has helped increase yield to a very large extent. Who is in India has not heard of green revolution, which was responsible for our country to not merely meet the national requirement in food production but also help us even to export it. Green revolution was dependent to a large extent on plant breeding technique for development of high yielding and disease resistant varieties in wheat, rice, maize, etc. 9.2.1 What is plant breeding? Plant breeding is the purposeful manipulation of plant species in order to create desired plant type that are better suited for cultivation. give better yield and are disease resistant. Conventional plant breeding has been practiced for thousands of years. Since the beginning of human civilization, recorded evidence of plant breeding dates back to 9,000 to 11,000 years ago. Many present day crops are the result of domestication in ancient times. Today, all our major food crops are derived from domesticated varieties. Classical plant breeding involves crossing or hybridization of pure lines followed by artificial selection to produce plants with desirable traits of higher yield, nutrition, and resistance to disease. With advancement in genetics, molecular biology, and tissue culture, plant breeding is now increasingly being carried out by using molecular genetic tools. If we were to list the traits or character that the breeders have tried to incorporate into crop plant, the first we would list would be increased crop yield and improved quality, increased tolerance to environmental stress, salinity, extreme temperature, drought, resistance to pathogens, virus, fungi, and bacteria, and increased tolerance to insects, pests would be on our list too. Plant breeding programs are carried out in a system way worldwide in government institutions and commercial companies. The main step in breeding a new genetic variety of a crop are first collection of variability. Genetic variability is the root of any breeding program. In many crops, pre-existing genetic variability is available from wild relation of the crop and preservation of all the different wild varieties, species, and relatives of the cultivated species, followed by their evaluation for their characteristics is prerequisite for effective exploitation of natural genes available in the population. The entire collection of plant or seeds having all the diverse allele for all genes in a given crop is called germ plasm collection. Second, evaluation and selection of parents. The germ plasm is evaluated so as to identify plants with desirable combination of characters. The selected plants are multiplied and using the process of hybridization, pure line are created wherever desirable and possible. Cross hybridization among the selected parent. The desirable character have very often to be combined from two different plants parents. For example, high protein quality of one parent may be needed to be combined with disease resistant from another parent. This is possible by cross hybridization. The two parents to produce hybrids possible by cross hybridization. The two parents to produce hybrid that genetically combine the desired character in one plant. That is very time consuming and tedious process, since the pollen grain from the desirable plant chosen as male parent have to be collected and placed on the stigma of the flower selected as female parent. In chapter 2, detail on how to make crosses have been described. Also, it is not necessary that the hybrid do combine the desirable character. Usually only one in few hundred to 1,000 crosses shows the desirable combination. 4. Selection and testing of superior recombinants. This step consists of selecting among the progeny of the hybrid those plants that have the desired character combination. The selection process is crucial to the process of the breeding objective and requires careful scientific evaluation of the progeny. This step yields plants that are superior to both of the parents. Very often, more than one superior progeny plant may become available. These are self-pollinated and several generations till they reach a state of uniformity, homozygosity, so that the characters will not segregate in the progeny. Testing, release, and commercialization of few cultivators. Newly selected lines are evaluated for their yield and other agronomic trait of quality, disease resistant, etc. This evaluation is done by growing these in the research field and recording their performance under ideal fertilizer application, irrigation, and other crop management practice. The evaluation in research field is followed by testing the material in farmers field for at least 3 growing seasons at several locations in the country, representing all the agroclimatic zones where the crop is usually grown. The material is evaluated in comparison to the best available local crop cultivator to check or reference cultivator. India is mainly an agriculture country. Agriculture account for approximately 33% of the India's GDP and employs nearly 62% of the population. After India's independence, one of the main challenges facing the country was that of producing enough food for the increased population. As only limited land is fit for cultivation, India has to strive to increase yield per unit area from persisting farmland. The development of several high-yielding varieties of wheat and rice in the mid-1960s as a result of various plant breeding technically to dramatic increase in food production in our country. This phase is often referred to as a green revolution. Figure 9.3 represents some Indian hybrid cross of high-yielding variety. Figure 9.3 some Indian hybrid crops: A. Maize, B. Wheat, C. Garden beans. Wheat and rice. During the period 1962-2000, wheat production increased from 11 million tons to 75 million tons, while rice production went to from 35 million tons to 89.5 million tons. This is due to the development of semi-dwarf variety of wheat and rice. Nobel laureate Norman E. Borlaug at International Center for Wheat and Maize Improvement in Mexico developed a semi-dwarf wheat. In 1963, several varieties such as Sonalika and Kalyan Sona, which were high-yielding and disease-resistant, were introduced all over the wheat growing belt of India. Semi-dwarf rice varieties were derived from IR 8 developed at International Rice Research Institute IRRI, Philippines, at Tachuk Native 1 from Taiwan. The derivatives were introduced in 1966. Later, better yielding semi-dwarf variety Jaya and Ratna were developed in India. Sugarcane, Saccharum barbarii, was originally grown in North India but had poor sugar content and yield. Tropical canes grown in South India but Saccharum africanum had thicker stem and higher sugar content but did not grow well in North India. These two species was successfully crossed to get sugarcane varieties combining the desirable qualities of high yield, thick stem, high sugar and ability to grow in the sugarcane areas of North India. Millets, hybrid maize, jowar, bajra have been successfully developed in India. Hybrid breeding have led to the development of several high-yielding varieties resistant to water stress. 9.2.2 Plant breeding for disease resistant. A wide range of fungal, bacterial and viral pathogens affect the yield of cultivated crop species, especially in tropical climate. Crop losses can often be significant, up to 20 to 30% or sometimes even total. In this situation, breeding and development of cultivators resistant to disease enhance food production. This also helps reduce the dependence of use of fungicides and bactericides. Resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. Before breeding is undertaken, it is important to know about the causative organisms and the mode of transmission. Some of the disease caused by fungi and rust, example brown rust of wheat, red rot of sugarcane and late blight of potato, white bacteria, black rot of crucifers and by virus, tobacco mosaic, turnip mosaic, etc. Methods of breeding for disease resistant. Breeding is carried out by the conventional breeding technique described earlier or by mutation breeding. The conventional method of breeding for disease resistant is that of hybridization and selection. Its steps are essentially identical to those for breeding for any other agronomic character such as high yield. The various sequential steps are screening germplasm, resistant source, hybridization of selected parents, selection of and evaluation of the hybrids and testing release of new varieties. Some crops varieties breed by hybridization and selection for disease resistant to fungi, bacteria and viral disease are released. Table 9.1 Conventional breeding is often constrained by the availability of a limited number of disease resistant genes that are present and identified in various crop varieties or wild relatives, including mutation in plant through diverse means, and then screening the plant material for resistance sometimes lead to the desirable genes being identified. Plant having these desirable characters can be either multiplied directly or can be in breeding. Other breeding methods that are used are selection among the somaclonal variants and genetic engineering. Mutation is the process by which genetic variation are created through changes in the base sequence within genes. See chapter 5, resulting in the creation of new character or trait not found in the parental type. It is possible to produce mutation artificially through use of chemicals or radiation like gamma radiation, and selecting and using the plant that have the desirable character as a source of inbreeding. The process called mutation breeding. In mung bean, resistance to yellow mosaic virus and powdery mildew were induced by mutation. Several wild relatives of different cultivated species of plant have been shown to have certain resistant character but have very low yield. Hence, there is need to introduce the resistant genes into the high-yielding cultivated varieties resistant to yellow mosaic virus. In Bindi, Abelmonsicum esculentus was transferred from a wild species and resulting a new variety of A. esculentus called Parbhani Kranti. All the above examples involve sources of resistant genes that are in the same crop species which has to be bred for disease resistant, or in the related with wild species. Transfer of resistance gene is achieved by sexual hybridization between the target and the source plant followed by selection. 9.2.3 Plant breeding for developing resistant to insect pests. Another major cause for large-scale destruction of crop and crop produce is insect and pest infestation. Insect resistance in host crop plant may be due to morphological, biochemical, or physiological character. Hairy leaves in several plants are associated with resistance to insect pests. Example: resistance to gesses in cotton and serious leaf beetle in wheat. In wheat, solid stems lead to non-preference by the stems solely and smooth-leaved and nectar-less cotton varieties do not attract bollworms. High aspartic acid, low nitrogen and sugar content in maize lead to resistance to maize term borer. Breeding methods for insect pest resistance involve the same type of those for any other agronomic traits such as yield or quality are as discussed earlier. Source of resistant genes may be cultivated varieties, germplasm collection of the crop, or wild relatives. Some released crop varieties breed by hybridization and selection for insect pest resistance are given in table 9.2. 9.2.4 Plant breeding for improved food quality. More than 840 million people in the world do not have adequate food to meet their daily food and nutritional requirement. A far greater number, 3 billion people, suffer from micronutrient, protein and vitamin deficiency or hidden hunger because they cannot afford to buy enough fruits, vegetables, legumes, fish and meat. Diets lacking essential micronutrient particularly iron, vitamin A, iodine and zinc increase the risk for disease, reduce lifespan and reduce mental abilities. Biofertilization Biofortification. In breeding crop with higher level of vitamins and minerals or higher protein and healthier fat is the most practical means to improve public health. Breeding for improved nutritional quality is undertaken with the objectives of improving: first, protein content and quality; second, oil content and quality; third, vitamin content; and fourth, micronutrient and mineral content. In 2000, maize hybrid that had twice the amount of amino acid, lysine and tryptophan compared to existing maize hybrid were developed. Wheat variety Atlas 66 having a high protein content that has been used as donor for improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing over 5 times as much as iron as in commonly consumed variety. The Indian Agriculture Research Institute, New Delhi, has been released several vegetable crops that are rich in vitamins and minerals. Example: vitamin rich carrot, spinach, pumpkin; vitamin C enriched bitter gourd, bathua, mustard, tomato; iron and calcium enriched spinach and bathua; and protein enriched beans, broad Love, French, and garden peas. 9.3 single cell protein SCP. Conventional agriculture produce of cereal, pulses, vegetable, fruits, etc. may not be able to meet the demand of food at the rate at which human and animal population is increasing. The shift from grain to meat diet also creates more demand for cereal, as it takes 3 to 10 kg of grains to produce 1 kg of meat by animal farming. Can you explain this statement in the light of your knowledge of food chain? More than 25% of the human population suffering from hunger and malnutrition. One of the alternate source of protein for animal and human nutrition, single cell protein. Microbes are being grown on an industrial scale as source of protein. Microbes like spirulina can be grown easily on material like wastewater from potato processing plant containing starch, straw, molasses, animal manure, and even sewage to produce large quantity, and can serve as food rich in protein, mineral, fats, carbohydrate, and vitamin. Incidentally, such utilization also reduce environment pollution. It has been calculated that a 250 kg cow produces 200 gram of protein per day. In the same period, 250 gram of micronutrients, microorganisms like methylophilus methylotropus, because of its high rate of biomass production and growth, can be expected to produce 25 tons of protein. The fact that mushrooms are eaten by many people in large scale mushroom cultivators grown industry makes it believable that microbes too become acceptable as food. 9.4 Tissue culture as a traditional breeding technique failed to keep pace with demand and to provide sufficiently fast and efficient system for crop improvement. Another technique called tissue culture got developed. What does tissue culture mean? It was learned by scientists during 1950 that the whole plant could be regenerated from explants. Any part of the plant taken out and grown in a test tube under sterile condition and special nutrient media. This capacity to generate a whole plant from any cell explant is called totipotency. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrients medium must provide a carbon source such as sucrose and also an organic salt, vitamin, amino acid, and growth regulator like auxin, cytoxin, etc. By application of these methods, it is possible to achieve propagation of a large number of plants in a very short duration. This method of producing thousands of plants through tissue culture is called micropropagation. Each of these plants will be genetically identical to the original plant from which they were grown. They are soma clones. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using the method. Try to visit a tissue culture laboratory with your teacher to better understanding and appreciate this process. Another important application of the method is the recovery of healthy plant from diseased plant. Although the plant is infected with a virus, the meristem, apical, and axillary is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plant. Scientists have succeed in culturing meristems of banana, sugarcane, potato, etc. Scientists have been isolated single cells from plant, and after digesting their cell wall have been able to isolate naked protoplasm surrounded by plasma membrane. Isolated protoplasm from two fused to get hybrid protoplasm, which can be further grown to form a new plant. The hybrids are called somatic hybrid. While the process called somatic hybridization, imagine a situation when the protoplasm of tomato is fused with that of the potato, and then they are grown to form new hybrid plant combining the tomato and potato characteristics well. This has been achieved resulting in formation of pomato. Unfortunately, this plant did not have all the desirable combination of the characteristics for their commercial utilization. Summary: animal husbandry is the practice of taking care of breeding domestic animals by applying scientific principles. The ever-increasing demand of food from animal and animal products, both in terms of quality and quantity, has been met by good animal husbandry practice. Their practice include management of farm and farm animals and animal breeding in view of high multi-nutritive value of honey and medicinal importance. There has been remarkable growth in the practice of beekeeping or apiculture. Fisheries and another flourishing industry meeting the ever-increasing demand of fish products, and other aquatic food. Plant breeding may be used to create varieties which are resistant to pathogen and to insect pests. This increase the yield of the food. The method has also been used to increase the protein content of the plant food and thereby enhance the quality of food. In India, several varieties of different crop plant have been produced. All these measures enhance the production of food. Techniques of tissue culture and somatic hybridization often vast potential for manipulation of plant in vitro to produce new varieties.